All right, everybody say hello.
Hello.
Say something that'll attract comments. We've cured herpes virus. Or you can actually survive on sunlight through a modified photosynthesis that we've developed for placing into humans. You know what you can't? No, really, you can't. All right, so we're finishing up our section here for the next exam, and it's going to be next week. I'm thinking Monday at this point, but I haven't completely made my mind up, and but I think we should plan that. And it's going to be the same way we did last time, where I'll drop it at 9, and you'll have 2 hours to do it, and it'll be very much the same kind of format and stuff. And I will put up, hopefully today, a list of the abbreviated lecture notes that we've got, and things like that. And in lab, we have a worksheet kind of on metabolism at the kind of level, at least to get you begin thinking about it in terms of what you need to know and how you should put this idea together. And we'll see where we go from there. Good. So let's just kind of recap a bit. I don't know if you saw the lecture, but we were going through the one anabolic metabolism pathway that we go through here. There are obviously lots of metabolic pathways. If you think about anabolic pathways, all these things we talked about of protein biosynthesis, the production of RNA, these are anabolic pathways. But a critical anabolic pathway is photosynthesis. And the way we kind of couched it is when we were thinking about an anabolic pathway, there are a couple of things that anabolic metabolism requires. One is anabolic metabolism is going to be a thermodynamically non-favorable reaction. And so that means we have to have an energy source, specifically in the form of ATP. We're going to need that phosphate to drive these various kinds of anabolic reactions, i.e., we're going to be phosphorylating enzymes or hydrolyzing ATP in a pocket, somehow making that protein change shape and probably push two things together. So that's one thing. Another thing that we can think about from our basic chart of metabolism is that these are going to be reduction reactions. And consequently, if reduction is a gain of electrons, we're going to need a source of electrons. And conveniently, photosynthesis provides both of these things. So we can break photosynthesis down into two distinct and discrete parts. There is what's known as the light-dependent reaction. And it used to be called a dark reaction, but we now call it the light-independent reaction because it can happen in the daylight. It just doesn't need light. So the light-dependent reaction, and that is what we have here on the board, the Hill reaction, which introduces electrons into what's known as the Z-scheme of electron flow. And then we have the light-independent reactions. And this is what's known primarily as the Calvin cycle. And there are a couple of different modifications to it, depending on if you're a cactus or if you're a corn plant. Can you see that? Yeah, well, close enough. It says light independent, light dependent. All right. So let's just kind of, as a review, going back through it in case you didn't see the video, and I'm always more charming in person, and so for you guys. One of the questions to ask is, where are these two things happening in this light-dependent reaction? And so I rather fancy myself of telling a nice story in that we start with photosystem 2 and photosystem 1 and finish up with the electron being picked up by the electron carrier NADP to become the reduced electron carrier NADPH, which is the supply of electrons, and asking the question, okay, if photosystem 1 gets its electrons from photosystem 2, where does photosystem 2 get its electrons? And the answer is, of course, the splitting of water. So this is what is known as the Hill reaction specifically. And water is split, and there's actually an enzymatic complex in photosystem 2 that is carrying out the splitting of water. And you do generate an oxygen radical for a time, but once you do a second water molecule, you make a molecule of diatomic oxygen. You can breathe that. Now, chlorophyll has the ability to absorb red light. And when it does, it kicks an electron into a higher energy state. Now, lots of things can absorb light and kick electrons into a higher energy state. But typically, the electron decays back to its ground state, and this is what we call heating something up. In photosynthesis, chlorophyll is actually a rather interesting molecule in that when it gets hit by a photon of light, it kicks an electron up into a higher energy state. And if we extract chlorophyll from a chloroplast and we don't have the ancillary proteins, which are essentially electron transport chain proteins, things like cytochrome, things that we see in the electron transport chain in mitochondria, if the electron decays back to its ground state, the energy is not released just as heat, it's also released as a photon of light in something known as fluorescence, which you have perhaps heard of. And fluorescence actually is a really cool phenomenon. What you can do is extract chlorophyll, shine UV light onto it, and it actually goes from being dark green to being a blood red because the light is kicking electrons up. Because they have no place to go, they decay back down. You lose some of the energy as heat, consequently, you have a longer wavelength, and then it also emits a photon of light. Now, in the intact chloroplast, that electron is kicked into a higher energy state, and then we effectively have an electron transport chain that is carrying out proton pumping. So we establish, i.e., the hydrogen ion gradient. And that is used to drive ATP synthesis. Now, because it is not identical to the mitochondrial electron transport chain, there are herbicides that can block this electron flow that don't really affect mitochondria very much. And so it ends up being a really good plant and photosynthetic plant specific herbicide because you're blocking electron flow, you block their ability to generate energy, and you kill plants. So there are a variety of herbicides that will do this. So once the electron makes its way to photosystem 1, which again is another chlorophyll molecule kind of neighboring in the membrane, surrounded by these other pigments, the light is, the red light again, slightly different wavelength, is absorbed, the electron goes up to a higher energy state, and now, depending on the balance of need, the electron can go into the electron transport chain again to generate more ATP, or it can be picked up. And if we think about this, this makes pretty good sense in that, you know, when you're making a bond, you need an electron. But in order to carry out all these different reactions in the Calvin cycle, you need a lot of ATP. So you probably end up having the ATP generating process going on a lot, and then every once in a while, electron will kick out and go into NADPH and get picked up. So this is the actual energy harvesting system. We've got the electron transport chain type system, and we have a proton pump, and we have an electron. Good. So just as a little microanatomy, comparing a chloroplast to a mitochondria. And if this was a plant physiology class, yes, we would go through the entire Calvin cycle and talk about all the different molecules. And it's actually a delightful path of interesting organic molecules. You take 3 carbons, there's a 4-carbon intermediate that can bind to make a 7-carbon. You have another 3-carbon that makes a 10-carbon. That 10-carbon splits into 2 5s. And it just is a really complex pathway that Calvin actually was able to elucidate really because they had access to radioactively labeled carbon, to C14 carbon. So this was carried out not long after World War II in Berkeley, actually in Berkeley, where they had access to the Lawrence Livermore nuclear reactor. And so they could generate radioactively labeled carbon dioxide, put into a tank with photosynthetic algae in the dark, and then boom, hit the lights on for a count of 3, and then dunk the whole thing into liquid propane to stop it, and then extract all the different carbohydrates and all the different carbon-containing molecules and run them out using something called paper chromatography, a fairly primitive system. But they were able to identify first the initial compound that was bringing in the radioactive carbon. And it is this 5-carbon ribose molecule known as ribulose bisphosphate. It becomes a 6-carbon molecule that then immediately splits into 2 3-carbons, and that is the beginning of the Calvin cycle. So a very complex, and it's something that is done all the time, all around us. Photosynthesis is happening everywhere you look, particularly now as spring is coming on. I'm looking out the window, it's bright green out there. That makes photosynthetic metabolism the most common metabolic pathway going on out here. It is said that if you were to take the entire world, extract protein from it in some sort of gigantic blender, the number 1 protein on the planet is ribulose bisphosphate carboxylase. Just a little trivia point. There you go. All right. So if we look at the ultrastructure of the mitochondria, and recall, I probably should be drawing this in green, for photosynthesis. Why not? Let's see if it shows up. We've got the standard burrito shape. It is a good bit bigger than a, yeah, you can see that. Sure, you're fine. It's bigger than a mitochondria. And it's got the thylakoid stacks stuck in the stroma syrup. So photosynthesis and chlorophyll specifically are actually happening in the thylakoid stacks. Now, one of the key points is protons, when they're being pumped, are not being pumped into the intramembrane space and being pumped out of this inner compartment. Protons are actually being pumped in, but the theory is the same. So the pH is lower in here than it is out here. So that was different, if you recall, and we'll get to that in a second. And we have a very similar ATP synthase hydrogen ion channel pump system that makes ATP. All right, so now if we were to compare this to a mitochondria, I'm drawing my mitochondria the same size. They're not quite the same size. I mean, we're talking about a Chipotle wrapped burrito versus a Twinkie probably. But you have this tremendous infolding of membrane. And if you recall here, Krebs cycle is going on deep inside in the matrix. And we drew that big diagram of electron flow happening on this membrane. And protons are being pumped out. Just for contrasting color, we have hydrogen ions being pumped out. And so we end up with a higher pH inside and a lower pH outside, the exact opposite of the mitochondria. But the electron flow system works just the same way. And in fact, you can interrupt it with uncouplers. We have in lab, there's an interesting article on dinitrophenols, DNPs, that are able to punch holes into the inner membranes. They probably work on chloroplasts, but nobody ever tries to do diet drugs on chloroplasts. All right, so lastly, we can begin to think about the flow of metabolites in the following way. Now, I kind of like to think of it this way as a comparison between plants and animals, but it really is a comparison between chloroplasts and mitochondria, because one thing that's important to recognize is that plants have both chloroplasts and mitochondria, which is a pretty cool trick. What this means is they're able to capture carbon, turn it into sugar, and then turn around and take that sugar, break it down with glycolysis in the cytoplasm, take the pyruvate, transport it to the mitochondria, pop that carbon off as acetyl-CoA, break it further down, and generate carbon dioxide and water. So they make their food from carbon dioxide, and then they consume it and generate more carbon dioxide. Pretty cool trick, if only we could do that. So let's just think about this here. The flow of metabolites between our plant friend here some sort of odd daisy looking character sitting in a pot. There's our plant carrying out photosynthesis. And now we've got our mitochondria, which we'll represent as some kind of little rat guy. Why not? There we go. So we have our animal over here. Now, the little fella here is taking in glucose and O2, and he's kicking out, by peeing on things, he's kicking out water and carbon dioxide. So this guy is breathing out and peeing on stuff. Now the plant is taking in CO2 and water, and it kicks out, at least the chloroplast this is, the plant is kicking out glucose in the form of starch or whatever, or plain sugar if you're a sugar cane, and O2. What do you think of that? We have a perfect match of what the mitochondria needs, the chloroplast provides. What the chloroplast needs, the mitochondria provide. A perfect match. And in fact, if you balance things out carefully enough, you can have a terrarium. So the easiest terrarium, and do we all know what a terrarium is? Where you take a mayonnaise jar and you put some dirt in it and some plants, and if you wanted to, you could stick a couple of little, if it was a big enough mayonnaise jar, a gerbil. You'll throw the gerbil in there. And you put it in the sunlight. So the gerbil is wandering around, chewing on the plant, peeing and exhaling. The plant is taking in the carbon dioxide and water, generating more glucose, more plant material. And as long as you have a nice balance, as long as the gerbil doesn't eat too much, you're going to be okay. Now, we actually are living in one gigantic terrarium here on our very own planet, in that this is, in fact, the cycle with which we're able to exchange nutrients between photosynthetic organisms and our mitochondria. So if you think about it, pretty much everything you're eating is either a plant or somebody that ate plants. And I guess if you're consuming things like tigers, you're eating something that ate plants. But generally, we don't eat carnivores. There you go. So this is the cycle that it works. And as long as you have enough space and enough sort of balance, you got to have a lot of plants and not too many gerbils. This works. And so there is, in fact, a pretty cool experiment that was done by Stanford out in the middle of the Arizona desert that was known as Biosphere. And I think there was actually a movie, Pauly Shore, who you may not even know, really dumb comedy movie based on it, but regardless. The idea was to take like 5 biospherians and place them into this thing that was a gigantic greenhouse, and it was completely sealed up. I mean, really took care to use lots of tape and caulk to make it so it was gas impenetrable. They established an oxygen-nitrogen atmosphere with some carbon dioxide, and they had multiple sets of biomes. They had sort of a dry area, they had a wet area, they had all kinds of plants growing in there. And then they took the biospherians, stuck them in there, locked the door, and said, all right, good luck, we'll see you in 2 years. And their goal was to see if they could, in fact, essentially harvest sufficient calories from their environment to survive. And the story goes that they got in there, and after a few weeks or so, began to realize that the caloric requirement was greater than what the plants could actually generate. And they had to go on a phenomenally restricted calorie diet. And they kept the experiment going for a while. The ability to make a pizza was like one of their stories of having to grow sufficient grain to grind into flour, and then getting tomatoes, and then getting sufficient milk from a goat that they could actually make cheese, and ultimately they could make a pizza. So we're lucky in that pizzas are easier to make on our planet in large scale, but it is a really neat kind of flow of metabolite. And lastly, it does bring about the fact that evolutionarily, we have to develop and wait for, life on the planet has to wait for photosynthesis to evolve, because the original atmosphere on our planet is mostly CO2 and nitrogen. And in that environment, all you can do is carry out anaerobic respiration. And there are a lot of bacteria that do some really weird things. Like, rather than using oxygen, they can use sulfur, or rather than using oxygen, they can use iron ions or nitrate ions, all kinds of things as the final electron acceptor. But oxygen has a really strong pull because it's electronegative. You get a lot better flow of electrons if you can use oxygen as your final electron acceptor, and you get a lot more efficiency in terms of generating this proton gradient. But you can get by with a range of things as the final electron acceptor. Now, when the photosynthetic pathway evolved, all of a sudden, the O2 concentration goes from being pretty much a trace. It goes up to where we are now at about 18%. And the nitrogen concentration is around 20%. The numbers are actually, 80% that is. It's really almost 20 and 80. Carbon dioxide and all the other gases make up really trace quantities. There is concern, and rightfully so, about increasing amounts of carbon dioxide, but the actual amount of carbon dioxide is actually fairly small. But as it increases, it changes the insulating ability of the atmosphere, and it leads to global warming. And that is a thing. And the number one way we deal with increasing carbon dioxide is to put more carbon dioxide consumers in there. Let's plant more trees and carbon fix by putting it into cellulose rather than trying to put it underground or I don't know what all. So the ability of photosynthetic organisms to convert water into oxygen and to begin bringing down the carbon dioxide is critical, because we now have sufficient oxygen that we can carry out oxidative phosphorylation. That is aerobic respiration. And that is the key to complex life, as we've been talking about it. In the absence of oxygen, your ability to make ATP from glucose, or your ability to rephosphorylate ADP, is dramatically reduced. You are running around 2 molecules per glucose, whereas in the presence of oxygen, you're doing between 32 and 36 molecules of ATP per glucose. And for a complex multicellular organism like myself, I need a fair amount of ATP. Consequently, I'm quite fond of oxygen. And that pretty much takes us to the end of the material for exam 2 and the end of the chapter. And I say, let's take a moment and, just for the heck of it, look at some pitches and see what we see. I went one too far. Nope, went too far. That's it. Chapter 14 is where we are. And we'll see if I missed anything. I didn't miss nothing. So how many of you have pet houseplants? Well, there are 4 people in the audience. Very good. Hopefully you treat them well, talk to them. Well, exhaling CO2 on them helps them. Increased CO2 concentration actually does help plants some. Light is their number one energy source. The other thing that plants are always limiting on is nitrogen. And it's always something to think about is where is nitrogen used in living cells and living systems, and number one place is in the amino acid. So plants are always hard up for protein, or hard up for nitrogen to turn into amino acids. So if you can give your plants some nitrogen, you're doing a good thing. So this is where we start out. And this essentially is the overall pathway that we just showed, in which we've got energy from food being turned into a hydrogen ion gradient in the process of oxidative phosphorylation. And in photosynthesis, we take energy from sunlight, and in photosynthesis, generate a hydrogen ion gradient. So this hydrogen ion or proton gradient is what we use to make ATP. So there are 2 different kinds of pumping systems here. We've got the electron transport energy is used to pump protons across the membrane. That's why having really impermeable membranes is so critical. And then that proton gradient is resolved as the protons make their way back through the ATP synthase, or as I say, the ADP rephosphorylator. And that is what's making ATP. So this system works. And it's actually a little bit like a battery. So just thinking about mitochondria, this is mitochondrial DNA is undergoing replication. The mitochondria actually do split. Mitochondria are being exposed to oxidative stress. They're being exposed to that oxygen radical. And so your mitochondria do get a little bit crispy and crunchy. And so there are lots of cells that are not dividing themselves, but inside the mitochondria are dividing and being recycled and kind of broken apart. And here we go. So there's the Twinkie, there's the Chipotle burrito. We've got the matrix, the inner membrane, the outer membrane, and a similar kind of architecture going on here. The main thing is the stroma, or the thylakoids, are generating the compartments that are going to be storing and generating proton gradient. So chloroplasts are a good bit bigger, but they're doing a lot more stuff too. Just as a little point, cells that are undergoing lots of respiration end up having, not surprisingly, a whole bunch of mitochondria. So if you want to harvest mitochondria, muscle cells are a really good place to do it. The tail of flagella are another good place, because we'll see later when we start talking about the cytoskeleton. Flagella and muscles just consume ATP like crazy as they are dephosphorylating it, pulling that phosphate off to change protein shape. And here we are, mitochondria. That's a lot of mitochondria. I'm not sure what cell that is. So here is the electron micrograph, in which we see the dramatic enfolding. That generates lots of surface area for the electron transport chain. And this is where, if you were watching Monday, or I made my edit, my production error, in that I just launched into my anabolic metabolism story before I really finished the 2 points of catabolic metabolism, and that was the fact that sugars and polysaccharides, they're going through glycolysis, and pyruvate, as it makes its way across the mitochondrial membrane, is turned into acetyl-CoA. And fatty acids and proteins, they're going to undergo beta oxidation, and 2 carbons are going in. I think the fatty acid really is not here, regardless. 2 carbons go in, and they become acetyl-CoA. And so that's how we're able to consume fats and proteins as an energy source. Yeah, so electron processing going on here. And here we go. Still not going to talk about proteins at all. Fine. If you can look at this diagram and understand what's going on and make comments about it, you're right on target. That's the level of detail we need to know. And, okay, there you go. Yeah, so this is the complex system. And rather than worrying about the names for now, when you start taking the MCAT, you can memorize them, or whatever. So this is complex 1. Complex 2 is not shown. Complex 3 is here, and this is complex 4. So we've got NADH dehydrogenase, which actually makes sense because that is the system that's pulling the electron off NADH, and turning it back into NAD. And then we've got the cytochrome system, and so the front guy is putting an electron onto cytochrome, so it's called cytochrome reductase. This guy is pulling an electron off of cytochrome, so it's known as cytochrome oxidase. And let's see, so here is ubiquinone, CoQ10, which is a supplement, is here. Complete nonsense. B12 is somewhere in here as well. Things like cyanide block the electron flow. So, if you're taking supplements, don't confuse your vitamin B12 with cyanide. That'd be really bad. And so this is the ATP synthesis system, or as I say, the ADP rephosphorylating system. And it is being driven by the proton gradient. So this is actually the protein complex that Peter Mitchell was working on that was being used to demonstrate that it was pH change that was driving ATP synthesis. And so one of the critical experiments he did was take mitochondria, and they did all kinds of, taking mitochondria apart and generating little vesicles and things. But if you take mitochondria and throw them into a weak acid solution, vinegar, don't put any glucose in, shut the oxygen off, nothing's going in, or you make ATP. And that was really the critical experiment to demonstrate that ATP synthesis is being driven by the proton gradient. Because you have to realize that that was an absolute bizarre notion of how ATP was being generated. And it was the critical step to understanding oxidative phosphorylation. As I also say, the term oxidative phosphorylation was generated prior to Mitchell's chemiosmotic mechanism. If we had a way of, sort of renaming, we'd call it the big proton kablooey or something, I don't know. But oxidative phosphorylation is not a very good term because really oxygen isn't involved. It is the proton ATP generating machine. But that's just me. All right. So this is now where you get into the nitpicky, when you start looking at the amount of ATP being generated by the different components of metabolism. And this is not something you need to memorize, but it is something really you want to make sure you can fully get the ins and outs of. So glycolysis, we have the net gain of 2 ATP. We're producing 2 NADH. Now, because they're in the cytoplasm, you actually have to use a little bit of ATP to transport them into the mitochondria where they can actually donate to the proton pump. So consequently, their yield is a little bit less than the yield once you're in the mitochondria. So 2 NADH in the cytoplasm gives you 3 ATP. 2 NADH if you're in the inside of the mitochondria, this is cytoplasm, gives you 5. But really, do we care? No, we do not care. So pyruvate oxidation to acetyl-CoA, we generate NADH there. The complete oxidation of acetyl-CoA is generating 6 NADH, 3 per turn. We also generate FADH2, thank you. And we also generate the 2 GTP molecules, which we count towards our ATP count because we're not picky. A nucleotide triphosphate of any flavor is fine with me. And just look here, FADH2, because where it starts in the mitochondria, in the electron transport chain, complex 2 doesn't have proton pumping capacity, so its contribution to the ATP generating system is a little bit less. 3 ATP per 2 FADH2 versus 5 ATP for NADH. So poor FADH2 is always kind of like the second-class citizen. Nobody likes FADH2. I do. Actually, seriously, it's like, why? It is used in a couple of other pathways, but you know, if you're trying to simplify things, really? All right. Some of the experiments that were done, blah, blah, blah. Anything good here? No. Anything good here? Not particularly. Redox potentials and calculation of delta G from the redox potentials. Oh, if this is what you're into cell biology for, really. So this is one of those molecules in the membrane that the electrons are passing through. My only main point is a fair amount of resonance structure. They can pick up electrons and lose electrons, no big deal. And so here is the energy pathway going on. And oxygen has got a really good drawing capacity. It pulls the electrons down. And there you go, fine, whatever. Really? No. I'm sure people know this, but do we care? No. All right, here we have some rocks. And I'm not sure if this is a slice of bread that didn't get mixed well or something like a chocolate pudding ribbon cake. I haven't had any breakfast yet. Now, what we're seeing here is over evolutionary time, photosynthetic algae and photosynthetic bacteria make the scene. And when they do, whoopee, we start to make lots more oxygen. This does look like some kind of nice chocolate swirl cake, but it's not. It's a rock. You can't eat that. So here we have chloroplasts. And, you know, they're green. This is a light micrograph. There are a bunch of chloroplasts in one cell. I don't know how many. I think we counted like 14 or so. We actually did a couple years ago a ratio-based real-time PCR, quantitative PCR thing, where we were looking at the DNA in chloroplasts and then comparing that to the DNA in the nuclei and able to make a ratio of chloroplasts to cells for different cell types. But there's a fair number of chloroplasts in a leaf cell. So here is our chlorophyll, or our chloroplast. We have banana there. And here are the grana stacks in the thylakoids. And here we have photosynthesis. So the light-dependent reaction, taking water, stripping the electrons off, generating ATP and NADPH, and then we go into the carbon fixation cycle, or the Calvin cycle. So this is what's known as the action spectrum. This is what chlorophyll actually absorbs. And just as a little note in spectrophotometry biz, those of you that are in analytical chemistry will appreciate this, that green chlorophyll don't absorb no green light. It reflects it or it passes it through. That's why chlorophyll and plants are green. They do absorb a fair amount of blue light and red light, but it is this red light that is actually driving photosynthesis. There are what are known as blue light effects, but there are some pretty weird things involved in flowering and stuff. So this is chlorophyll. And as we can see, A, it's got a lot of electron resonance possibilities. B, it is extremely hydrophobic. It's stuck in the membrane, deep in the membrane. Now, why they have it blue, not green, I don't know. So we got the photosystems and the reaction center. All right, so this is what we were drawing more or less with photosystem 2 passing the electron that it extracts from water into this electron transport chain. It's actually a fairly simple electron transport chain. You only got one proton pumping guy, but regardless, it's pumping the protons into the thylakoid space, and so this is the syrup, the stroma. The pH is higher outside than it is inside, so I guess you could, it's fortunate that mitochondria do it the other way around. Otherwise, that dunking it into acetic acid or into vinegar experiment really wouldn't work. I was thinking you could maybe dump this into like baking soda, but you might not have enough hydrogen ions to make this thing go. So fortunately for Peter Mitchell, chloroplasts are just weird, and he was looking at mitochondria, not chloroplasts. And we got actually a very similar ATP synthase system that's taking the hydrogen ions and moving back through. All right, so photosystem 1 is where we're taking the electron and putting it into this final electron acceptor of NADPH. So they're drawing them as two separate little spots. I kind of like my model better. Nothing good here, move on. So here it is in, rather than sort of the electron energy, just in a line, which is how it is in the membrane, let's face it. You have photosystem 2 passing electrons on to photosystem 1. Photosystem 1 is then taking that electron and putting it into NADPH, and we make ATP.
So there's a higher pH inside the ground?
There is a higher pH comparatively outside the thylakoid membranes than inside. So the protons are being pumped into the thylakoid membranes. So if we think about, if we kind of maintain our pancake analogy, we're putting protons into the pancake, whereas if we can think about our burrito system, we're pumping protons out of the burrito in the mitochondria. All right, so here we have the electron flow, and at some point we're going to get into cyclic photophosphorylation.
So then mitochondria, there's a higher pH
Outside.
So then they're both like outside?
Oh, no. Wait, higher hydrogen ion. Sorry. It is a higher hydrogen ion concentration outside. It is a lower pH in mitochondria. What you said was correct. So in the mitochondria, we're pumping protons out. In the chloroplast, we're pumping protons in. And, here we're getting actually to the Calvin cycle. So we have carbon dioxide, ribulose bisphosphate. And what I think is quite amusing is, your goal is to make glucose, 6-carbon. And I can only imagine that when Calvin did the initial experiment, and you say, okay, we're going to take radioactive carbon dioxide, and we're going to put it into this photosynthetic algae mix, and then turn the lights on for 1 second, and then dump it into liquid propane. It's like, bam, boosh. And then you extract the carbohydrates from this thing, you run it out, and hey, we've got a 6-carbon molecule. It's glucose. I'm a hero. And then you say, okay, now let's run it for 3 seconds. And now, where'd all the glucose go? Because it ends up being split into 2 molecules of phosphoglycerate. So, no, it wasn't that simple. If it was that simple, it wouldn't work, and B, he wouldn't have gotten the Nobel Prize. This is the carbon cycle. Pretty cool. And at one point in a near graduate level plant physiology class, I had to memorize this whole thing. Don't worry about it. All I know is that there are some molecules in here that we as animals do not even come close to producing. There is a 7-hepulose 7-phosphate floating around in here, which is a 7-carbon sugar. And man, you ain't got no 7-carbon sugars anywhere in your body. So there you go. Photosynthesis is pretty darn bizarre. One turn of the Calvin cycle results in the production of, or the fixation of 1 molecule of carbon dioxide. You turn this 3 times, and you make a 3-carbon glyceraldehyde 3-phosphate. You accumulate that, and that's what goes on then to start doing all kinds of sugars, fats, amino acid production. Of course, with amino acids, you also have to put the nitrogen in there too. That's why you fertilize your plants. And here is an electron micrograph of a chloroplast. So in this case, the protons are accumulating in the thylakoid stacks in the grana, and that's where the lower pH is, and so the protons are moving from here out. And last but not least, we've got the flow of metabolites from the mitochondria to the chloroplast. I still prefer my plant and rat, but let's be fair that plants have both. Plants are pretty much a self-contained machine. If people and all animals were to vanish from the planet, some plants are going to make it. I guess if you need a bee to pollinate, all right, regardless. If people were to vanish from the planet, plants wouldn't care. They're able to operate in a closed cycle, in a closed system, pretty darn well. So this is another one of those fundamental conceptual thingamabobs that if you can get this, you're on target, you're doing good. Yeah, these are some of the experiments they did, blah, blah. All right, so this is a timeline starting a while ago. Okay, 4.6 billion years ago, Earth forms. First living cells around 3.6 billion years ago. Pretty quickly, photosynthetic organisms make the scene, and we have now the first water-splitting photosynthesis, but it takes another billion, or another 2 billion years to actually get to where we have enough oxygen starting to accumulate that aerobic respiration evolves. And finally, we now have eukaryotic photosynthetic cells that continues to crank the oxygen production. And here we are with the first vertebrates coming up. First multicellular plants and animals are a little bit before that. First vertebrates are here, and that's you right there. So we can thank photosynthesis for life on this planet. Yeah, just some weird guys that aren't carrying out metabolism. There are creatures that live in volcanic vents deep under the ocean, and they don't have a lot of oxygen. They're carrying out some strange metabolism, and it's one of the things that, as I've been doing microbiology, there are these accepted ways we think about plants, or we think about animals. This is what we are. This is how we eat. This is how we operate. And then there are some really weird ways of operating, of using other kinds of inorganic ions, all sorts of stuff as the final electron receptor. That's enough. That's all I have to say. Anything more good here? Good. In slideshow. Hey, I'll catch up with some of you tomorrow, and we will continue on the road. I'll make the final determination for the exam, because I do want to spend Friday going over membrane biochemistry, active passive transport. So, yeah, I think Monday is going to be the exam. Let's go ahead and plan on that, and if something happens or if you can't stand that, holler.